Good morning, everybody. Welcome to Bioinformatics. It's day 1 of 5. In past years, we've used as many as 8 days for teaching this subject. This year, we have 5. As a result, I've compacted some material out of past years. So things like binary, hexadecimal, and photographic representation in bytes is not going to be included in this year, but that stuff is all available on YouTube videos that we've produced in the past. So if you end up doing microscopy and want to think about ways to do image analysis on it, those slides are also available through YouTube. Okay, so today we're going to start with some of the things that we need to know about working with sequencing data or with sequence variant microarray data in order to do things like GWAS. So we're going to be talking about the detection of variance from DNA sequencing data. I realize, however, that people may have relatively little information about how sequencing works. Has it been a while since you all went over things like di-terminator sequencing? All right, so we'll have a little bit of review on that and try to compare how old and new sequencers differ on the score. And from that, we're going to talk about 3 big bioinformatics areas. One of these is Phred, which is a technology that's been around for quite a long time. And even if we don't use the original software to produce Phred scores, the concepts behind Phred scores are very much alive. So we're going to be talking about that. From there, we will talk about the fine exercise of mapping. One of 2 major bioinformatics exercises that we use in the space of next-gen sequencing. So we'll be discussing Bowtie as a result. And then from there, we will talk about GWAS, or Genome-Wide Association Studies, particularly with relationships to the terms of linkage and admixture, a bit of stratification as well. Just so you know, the quizzes that I tend to write focus around the key terms. So along the way, if you see a term that I'm laying particular emphasis on, you might just make sure that in your notes you can write a succinct definition of it. That will get you past about 70% of the questions on the quizzes. So I want to be sure that when you encounter these terms, you have a working definition to go by. Bioinformatics is not one thing. I very frequently see people who don't know a lot about bioinformatics writing course designs that are all about sequence alignment. And sequence alignment is nice. It's a great technology, as we'll discuss at some length tomorrow. But sequence alignment is not the end-all be-all of bioinformatics. So as we look at this diagram, we can see that all of these different pipelines start with, okay, so everybody should have their phones silenced. I'm not sure who's getting text messages, but go ahead and shut off the noise on your phone now. So you can see that we have all kinds of different information pouring out of genomics and transcriptomics. Now, I, as a protein biologist, feel awkward about the fact that we're not talking yet about mass spectrometry and so on. That will come later in the week. I believe that's on Thursday, so we'll be getting to that later. But for now, I want you to think about data flowing from sequencers here in high-throughput sequencing, maybe genome-wide sequencing, or even through exome sequencing, where we're focusing just on the genes that get expressed in the genome, or even dealing with data that talk about SNPs, popularly observed or frequently observed variants that are frequently used to track heredity, for example, in populations. So you don't have to have a sequencer for some of these things we're talking about to be relevant. And of course, once you get to transcriptome, once you're talking about things like gene expression, you're probably dealing with microarray data or with RNA-seq. We'll talk about that in some depth on Wednesday. But today, the track that we're really focused on is this process of base calling, read mapping, variant detection, and phenotype assessment, which variants actually matter, along with things like admixture and genome-wide association studies. So that's kind of the roadmap for where we're going to go, just straight down that column today. All right, I've given myself 90 minutes to cover all these topics, and I always feel like I get myself in trouble when I have a nice open-ended slide like this. But I hope everyone recognizes this as the structure of a nucleoside. This is one side of a DNA molecule, or it could be in this. It could be using a 5-carbon sugar. It could be a ribose, an RNA instead. So we see that we have a backbone that runs phosphate, sugar, phosphate, sugar. We also see that these carbons on the sugars have been labeled. You see that? 1, 2, 3, 4, 5. So when we talk about 5' and 3', we're talking about directionality in a DNA strand. So when we build DNA, we are always adding onto this 3' hydroxyl that comes off of the 3 carbon on the sugar. That becomes the point where we attach a new nucleotide to this. And the bit that we talk about all the time with A's, C's, G's, and T's, those are represented by the nucleotide, these nitrogen bases that can form hydrogen bonds among themselves. So a little thought about that structure gives us some clues about how sequencing can proceed. So as was voiced in the very first paper on DNA structure from Watson and Crick, the structure of DNA lends itself to copying by the nature of its structure. Because if you separate the 2 strands of DNA, each of those is a perfect template for recreating the other side. So unzipping the molecule and then polymerizing a complementary strand is a pretty straightforward kind of operation to do. So when we do sequencing, we are also generating complementary sequence. We're still relying upon polymerization, but we're also putting in some points of failure when we're doing Sanger sequencing, because it is possible to polymerize in a nucleotide that does not have an attachment point for any further nucleotides. Right? So if it doesn't have that hydroxyl there, you've got no ability to add more nucleotides after that point. So if your sequencing mix has mostly normal nucleotides, but some fraction of dideoxy nucleotides, anytime the complementary strand is being built and it hits a dideoxy nucleotide, it can't build any further. So you might start with a template and then polymerize out a whole bunch of complementary strands, but some fraction of those are going to stop after, say, 10 nucleotides. Some fraction are going to stop after 11 nucleotides. Some will stop after 15. And so you have this variety of complementary strands that have been built. Here we see these colored letters represent our dideoxynucleotides. So we see that our complementary strand may range in different lengths. Those can then be separated, often in capillary electrophoresis, and you can simply say which fluorophore appears at each position in this separation, and that tells you what the sequence is. Do people get that from Sanger? Okay. It used to be done prior to the use of fluorophores using radionuclides. That's not a technique that we use a lot now, but it was the separation and the dyed terminator aspect of it are very common features of anything that we call Sanger sequencing. Now, I want to stress that this is not how we typically do sequencing today. So, among other reasons, if you have to work from a purified template like this, the number of different sequences that you can produce complementary sequences for in one go is limited to the number of separations that you can do in your instrument. And that's typically limited to about 384 for like the deluxe model, right? And in the old days, having 24 capillaries was pretty big. But in the days of the genome sequencing project, having, say, 384 different capillaries was possible. But that is not what we do these days when we send things out for sequencing. Most generally, we are using what a lot of people call next-generation sequencing. I hate that term. The reason why is that next-gen sequencing has been a thing for more than 15 years, and the first next-generation sequencers would seem positively primitive today. So I really prefer the term massively parallel sequencing because it describes more accurately what these instruments are doing. And the Q is right there in the name. They are massively parallel sequencers because you're sequencing a tremendous number of templates all at the same time. We're no longer limited to being able to do 96 or 24 or 384 different sequences at a time. We're able to do millions of sequences. Millions of different templates are all getting sequenced simultaneously in each run of the experiment. That is pretty remarkable. So I want to remind you of how cartoons work, right? When we're dealing with a cartoon show, we're dealing with somebody having drawn or computer generated different cells of the characters, right? So if you were to pull out one frame of an animated movie, you would see the characters frozen in mid-sentence, right? But then if you look at the next frame, you're going to see that mouth has moved just a little bit, and the arm maybe has swung down, right? So our eyes are tricked into seeing this motion by repeatedly replacing the image that we see on screen. In the same way, we have a time-resolved signal that we're receiving from many different templates, all being sequenced at the same place. So let us imagine that our massively parallel sequencer has relatively few templates taking place, being sequenced all at the same time. Here we just have 4 different sequences that are going to be sequenced simultaneously. When we incorporate the very first nucleotide, we see that a T is incorporated in the sequence at this top position, a G is incorporated at the left position, a C is incorporated at the bottom center, and an A is incorporated at the bottom right. When we look at time point 2, we see that we have a different letter in each of those positions. We infer that T is followed by A in the sequence at the top center. It's then followed by an A, it's then followed by a G, it's then followed by a T. So we infer that this sequence starts with T, A, G, T, because we have different flashes of nucleotides being incorporated serially. Simultaneously, at the very same time, just on a different location on this bed, we had a G incorporated in the left sequence, followed by a C, followed by a T, followed by a G, followed by an A. And so we see that the software has extrapolated out that the sequence of this left template is G, C, T, G, A. Because we have the ability to resolve millions of different spots all in this bed at the same time, we are able to track which color gets incorporated at each location of this bed simultaneously for millions of templates. If you think about it, that is kind of an astonishing thing. But I would also point out that you carry amazing technology around in your pocket, most of you. A couple of years ago, I bought myself my first Android phone, and I was so thrilled to have a 10 megapixel camera that was built into the camera. I mean, 10 megapixels. Who could possibly need more resolution than that, right? If you think about it, that reflects 10 million different locations in a 2D plane. The sequencers have the ability to read fluorescence from a much greater resolution than 10 megapixels because they're routinely sequencing 10 million things at the same time. So you can imagine that the resolution required to be able to unambiguously match a letter from this moment in time to a letter from that moment in time at the same location. Okay, so that's the difference then from Sanger sequencing, the old dye terminator technology, and massively parallel sequencing or next-gen sequencing. You will not be punished for calling it next-gen sequencing. Okay, there are lots and lots of bits of jargon that come along in this world, and before I just lurch ahead, I want to be sure that we talk about some of them. One of these is template. I've already mentioned that. We frequently refer to as a template this sequence that we're inputting. What is the strand that we're putting at a particular location on this bed to have complementary sequences built for? Okay, so the template is the thing that we're going to sequence. And in massively parallel, we have millions of templates. Next, a read. A read is a really important bit of language. It shows up all over the place, so make absolutely sure you know this one. A read is a set of sequential letters that correspond to the complementary sequence of a template. So it's one individual sequence out of this. If you have if you have 10 million templates, ideally you would have 10 million reads produced from them. Shotgun sequencing is a term of art. It mostly refers to a random process that you have these millions and millions of templates that are being presented to the instrument for sequencing, and they're frequently drawn at random from some genome, right? So it may be that you're doing a kind of a survey sequencing of the alligator, right? We need to know what the sequence of alligators are, right? It's important meat if you live in the south of the United States, where I do. So if you live in Florida, you want to have the sequence of alligators, and you can do something light, like a 2x coverage of the alligator genome, scattering reads everywhere throughout its genome. And later, you hope that you can pull that information together in a way that lets you get to chromosome scale assemblies. Now, I said 2x coverage a moment ago. That's a reference to fold coverage, which is to say that we have some number of known nucleotides in the alligator genome, and we are going to do enough sequencing to see every nucleotide on average twice. Now, does that mean that every nucleotide in alligator is observed twice? Absolutely not. Because when you produce sequences at random from a genome, you're always going to have some places in the genome that end up being unexpectedly popular and some parts that are just unexpectedly ignored, right? So a random 2x coverage of a genome does not generally give you a very highly contiguous genome. You have a whole lot of gaps that result in a case like that. Generally, if you're doing de novo sequencing of something, you want more like 50-fold coverage, nice deep coverage, and you'll still end up with gaps even with that high a fold coverage. So what differs between the old school approaches with Sanger sequencing and massively parallel sequencing? First off, longer reads. In the old days, our sequencers could give us up to 600 nucleotides for each template sequence, right? That is a pretty long read. By comparison, most of the massively parallel sequencers out today generate very short reads. You can see here that I've listed 150 base pairs. That's only a quarter the length of what the old Sanger technologies could give us. So you might think, well, new technologies are better, right? I mean, we start with that assumption. If you buy a phone this year, you expect it to be better than a phone you bought last year. Well, you shouldn't waste your money on a phone every year, I think. But at least I always feel embarrassed about giving up on a phone after only a year. So in the same way, you might think that just because you have a massively parallel sequencer, it's better in all respects than Sanger sequencing. Not true. Most modern sequencers produce shorter sequences than do the old ones. That is not universally true, however. There's an instrument by a company called PacBio that will produce very long reads by comparison, much longer than what you could get from the old school Sanger. So this is a common rule, especially for things like Illumina sequencers, but it's not universal. Sanger sequencing generally produces more accurate base calls. So we have a read that reflects the letters that we got out of our sequencer, and we're now going to talk about the quality of individual letters within that sequence. So you got back 150 letters. Which ones can you trust? Which ones should you throw away? Is it possible to include or exclude a read based on the quality of individual letters and so on? So in general, we see that Sanger generally does a pretty good job of sequencing each template that you give it. You don't have that same kind of recall from the templates that you give to massively parallel sequencing. And of course, the big sales pitch, massively parallel sequencing is valuable because it is able to produce sequences from an incomparably large number of sequencing templates. Any questions? I thought I saw that. Okay. Feel free to poke in a question, because my goal here is that you guys understand what we're talking about. If any of this is going right past you, feel free to stop me. Okay. So we will also be spending some time talking about microarrays. There's a large number of people who again say, gosh, microarrays were so hot in the 1990s. Does everyone remember the 1990s in this room? Was anyone born mid-1990s? Don't make me feel too old. Oh, it's too late. All right. Well, I was born in 1973, so I was in graduate school in the 1990s. All right. So it may be that you think of microarrays as this ancient technology that no one would be bothered to think about. But in fact, just like Sanger sequencing, microarrays have stuck around. We're going to have a lot more to say about them on Wednesday. But you can use microarrays not just for gene expression studies, but to understand which genetic variants people have. So one of the big projects that kind of dwarfs a lot of other things going on is this H3 Africa program. H3 Africa has been designing bead-based array studies, a bead-based array strategy that allows us to evaluate genetic variation in African populations. So there's a lot going on in microarrays even now. They are certainly much cheaper to use for getting sequencing data, and they're also a whole lot easier to process on the back end. So microarrays are not dead, they're still very much viable. And I would just point out that at present, sequencing a genome still costs something like $1,000, 15,000 Rand, 14,000 Rand, something like that. Whereas running a microarray or beta array for genetic variants usually costs about 1/10 as much. So if you don't need sequencing data, it certainly makes grand sense to spend your budget on arrays rather than massively parallel sequencing. Okay, which brings us to electropherograms. Now this concept might seem a little weird if you have never seen one before. This represents an old school sequencer. This is like a Sanger sequencer. And I want to explain how the information collected by this instrument relates to the base calls that you see in the reads. So I'm going to point out that in this diagram, we have green peaks that correspond to A's, we have blue peaks that correspond to C's, red peaks that correspond to T's, and blacks that correspond to G's. So if you see a green coming up and down all by itself, you say to yourself, that green corresponds to an A in the sequence at this position. Now, if you look at this area and this area and this area, you probably say, well, it's pretty clear, I could read that. You know, I've got, okay, red, red, green, 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 black, green, 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 I'm sorry, red, red, red. You get the idea. Not that hard to just read these and interpret them directly. However, I would point out that there are some regions of this electropherogram that are quite a lot messier. So look at this part up front. All right, so I think most people realize that the separation that's taking place in the capillary electrophoresis is doing an absolutely lousy job with all these products up front. It's got, the dye signals are saturating the detector, and it's not separated at all. So at some point, we switch over to well-behaved electrophoresis, but that very first part is trash. Down here at the bottom, we see that we've got pretty good separation in, say, the 300s, but dip down into the 400s, and we no longer have what we call baseline resolution. Have people heard that term? In other words, up here, when green is up and then it comes back up again, it falls all the way down to the baseline. So that's baseline resolved because the signal goes all the way back down to 0 before it comes back up again. Once we get into the 400s, here we have this little run of A's. We see that it's not dipping all the way down to the bottom again. So we're no longer baseline resolved. We can tell there's a peak here, but it's not as clean as if we had baseline resolution. And by the time we get into the 700s, look at this mess. Could anyone really tell how many different green peaks there are around this region of 720? That's pretty bad. So we want to have information about each letter, about each base call, that tells us how much we can trust it. And that is what Phred is for. Phred was created by a team under a guy named Phil Green, who was actually on my PhD committee at the University of Washington. It's a pretty amazing piece of software, and I don't feel that I can tell you everything about it, in part because of time, so we're going to move through it pretty quickly. But Phred has persisted over time as one of the dominant ways that we evaluate the quality of individual letters, individual base calls in a read. Very important to know that part. So we have to infer some sequence from the electropherogram, and it's not going to be you sitting there saying, okay, red, 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 green, green, blue, black, black, green. Not going to do that. We're going to do that with software. And then Phred is going to give us an evaluation of whether those base calls are trustworthy or not. So a good base call should have these properties. A peak matches the beat of its neighbors. And I put beat in quotes here. So I want you to imagine that you're at a friend's party. You're celebrating the end of class period, and you're having an enjoyable beverage, and the music is on, right? The music is on. If the friend hosting the party is an audiophile, you probably have an excessive amount of bass, right? So you can imagine the beat is going, right? So you can imagine the same thing when we look through these electropherograms. We have a thump, 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 thump that is reflecting the increase and decrease in amplitude as a function in time. And so these electropherograms have a beat that we need to be able to evaluate in our software. All right. Next, only one trace is concave down at this call. Does that term make sense? Does everyone remember what concave looks like? Somebody use a hand to show me what concave looks like. That's good. That's good, yeah. So here she's describing a curve, thumb in the hand here. So we have this space right here. This is concave. But if we were looking at the outside of the hand, we would be looking at convex, right. So we're going to ask about the concavity of the signal, and it needs to be concave down. So I want you to point downward. So at this point, we're saying if we're going to call this a base call, we want the signal to be concave down. Now, if you're do we have math people in the room? We have a few math people in the room. We have at least one. Math people. You can admit it, it's okay. I was on math team in high school. I love this stuff. All right. Well, do you remember second derivatives? Somebody took calculus here. Somebody? Okay, good. I didn't see anyone wince when I said calculus. That's fine. All right, so the second derivative gives us a way to evaluate mathematically whether something is concave down. All right, so we want the signal to rise and fall as a peak. I mean, you look at Devil's Peak, you see something come up and down. You know what these things look like. All right, finally, highly probable errors are far away. Now, I moved to Nashville in 2005. Nashville's become a ludicrously popular city since that time. But in 2005, there were a lot of neighborhoods that were in the process of gentrification. They'd been where real people live, and then people were starting to move into town to live in these hoity-toity artist community neighborhoods that artists could no longer afford to live in. So the house that I lived in had been flipped. And in the United States, there's a lot of old properties that enterprising developers purchase at a low price, dump a bunch of money into, and then sell at a high price. The house I moved into had been flipped. And soon after arriving, I learned that this neighborhood I lived in, 12 South, used to have a drug problem. And the drug problem was nowhere else than my own house. My house was the drug dealer house. And it had been turned into this beautiful mini mansion, and it was gorgeous. But 5 years previously, drugs. Drugs, right. So when you think about which houses are expensive in a neighborhood and which ones are cheap in a neighborhood, you probably think about where is the crime. If you are next door to the drug house, your property values go up or go down? Down, right. Having a drug dealer next door, bad for property. On the other hand, if there's no crime reported in your neighborhood for 10 years, your property values are probably pretty high. In the same way, in this electropherogram, if you happen to be near something like this, where you've got two peaks that are kind of interfering with each other, being close to that makes all these neighboring peaks more suspicious, whereas something that's relatively far away doesn't bear that taint. See that? Okay, so now that we've discussed crime statistics and parties from finals, let's talk a little bit about math. Anytime we talk about beats, being able to recognize how frequent something is, in the back of your mind you should think, I'll bet Fourier Transform has something to do with that. Fourier Transform. When you go to your friend's party and they have a graphic equalizer, you may see that at the left edge of the graphic equalizer, every time there's a bump that you hear in the sound system, there's a big spike that comes up in that leftmost channel. And every time the lead guitarist goes into a solo where he's got his hands vibrating very close to where the frets are being held down, you get these really high squealing notes pouring out of the Stratocaster. You see motion not at the left edge of the equalizer, but at the right. You see spikes showing up there. That is because that equalizer is dissecting the sound currently playing into frequencies. And the low frequencies correspond to this bump, bump, bump. And the high frequencies correspond to the Stratocaster. Okay, so Fourier Transform is the way that we transform these time-dependent signals into frequencies. So anytime you hear Fourier Transform, you should think to yourself, there's a frequency detection taking place. But here, instead of looking at amplitude as a function of time, what we think of as sound, we're looking at signal intensity, fluorescent intensity, as a function of separation time. Okay, so Fourier Transform represents a way to get frequencies out of a time-dependent signal. All right, so let us imagine then that for our chromatogram, we're looking just at these 4 peaks for the moment. We have these 4 locations that have been predicted based on Fourier Transform. The Fourier Transform says the period should be exactly this for this part, of the signal. So we should assume in this case that these periods are exactly the same width in this case. But the peaks that we observe are not a perfect fit for those model locations. Some of them are scooted a little bit left of where we thought they would be. Some of them are spot on. In some cases, they're not even all that well shaped, right? Especially as we get to very low or very high peaks. So we have this shift between the predicted and observed location. And our understanding of whether this peak is reliable or not is going to hinge on how well it conforms with what we expect of well-behaved electropherogram peaks. And now we get to the piece that everybody should know about this. So when we talk about negative log scoring, we're talking about something that shows up in bioinformatics all the friggin' time. All the friggin' time. So I would say one of the things that we must do a lot of times in bioinformatics is differentiate between very small numbers that differ in magnitude. Let us imagine that we have probabilities. In this case, the probability we're going to assess is how likely is it that this base call is an error. Now, if something has a 50% chance of being an error, there's very little chance we're going to give it any further thought in this analysis. What we care about more are all these probabilities that are very close to zero. Something that has a 10% chance of error is quite different than something that has a 1% chance of error. Something that has a 1% chance of error is different than from something that has a 0.1% chance of error. So by using the negative log scale, we are giving ourselves the ability to differentiate between very small probabilities. Now, the math for this is log base 10 of the probability of error multiplied by negative 10. Why do we do that? Well, first off, this is a log base 10. So this log base 10 then is pretty easy to map. So a moment ago I was talking about percent chances of error. If I have a 1% chance of error, that's the same thing as 0.01. Everyone remember that? 1%, 0.01. There it is. In scientific notation, you would write that as 1e minus 2. Does everyone remember that from high school? 1e minus 2. Okay, so just grab this minus 2 part and multiply that by negative 10. Minus 2 times negative 10 is? It's 20. So a quality score of 20 is the same thing as a 1% chance of error, is the same thing as 1 in 100 of these being bogus. A 1 in 1,000 chance of error is 0.1%, which is the same thing as 0.001 right here, which is 1e to the minus third. So grab this exponent, minus 3, multiply that by minus 10, out pops 30. If you have only a 1 in 10,000 chance of error, you have 0.0001. That's the same thing as 1 times 10 to the minus fourth. Minus 4 times minus 10 is 40. Okay, so the relationship that we're drawing out here is that we have relatively small changes in the Q value that represent orders of magnitude more confidence in a base call. If you have a base call of 20, you're probably likely to throw that out. That means that there's a 1% chance of error at that location. If you have a base call of 40, your chance is much lower that this is bogus. But I want to contrast this with something that's going on in a massively parallel sequencer. You're sequencing millions of templates. Millions of templates. So events that happen 1 in 10,000 times happen a lot when you have millions of templates. A million is what in scientific notation? 1 times 10 to the 6th, exactly. So if you have a million events, something that happens 1 in 10,000 times happens how many times? In a million events? Okay, let me do this a different way. I'm asking you, if I have an event that happens 1 in 10,000 times, how often does it happen in a million events? So what I want everyone to do is write down the number 10,000. Everyone have that? How many zeros in 10,000? 4, yes, right, there are 3 in 1,000. So we're writing the number 10,000, so that's a 1 followed by 4 zeros. How many zeros do you have to add to get to a million? 2. Okay, so that's your answer right there. The difference between 10,000 and a million is 100-fold. So if you have an event that happens 1 in 10,000 times, and you have a million events, 100 of them are likely to take place in that space. So rare events, like having an error in something with a quality score of 40, can be not very rare at all when you're comparing it by many, many sequences. Okay. Now, one of the things that we frequently want to do is evaluate whether the money we invested in our sequencing experiment was worth anything. So one of the things that we often do is evaluate the quality scores that occur across these sequences. Now remember, we're not making a quality score for the whole read. Our quality score is built around this base call, this particular letter in the sequence. So you can imagine, as you saw in the electropherogram a moment ago, that the sequence calls at the very beginning of the sequence and the ones at the very end of the sequence may be questionable. So one of the things we can do is ask, what is the typical quality score as we range across this read, or across this collection of millions of reads when we're doing it this way? So here we're comparing across many, many different base calls. Here we have a sequencer that's outputting 40 letters in each read. So these are really short reads, like the old school Illumina stuff. So we can ask, what is the typical quality score when we look at the first nucleotide or when we look at the last nucleotide? And you can see that in the extreme cases, as we get to higher and higher base positions within these reads, we get to more and more questionable results. So this is one of the things that we do to evaluate whether the experiment worked well or poorly. FASTQ is a relatively quick way to get to that. All right, we still have a lot of material to cover, so I'm going to just try to blast ahead. I hope that if you run into questions, you're going to ask me. So I'm going to pause and look at you really searchingly. Oh, you're just moving your hair. Okay, I thought it was going to be a good question there. I was sure you had one. No, no question at all? Oh, all right, well, I'm waiting for it. I'm going to try to keep my eyes on you. So, what can you do with output from a sequencer? We have two chief uses that we use, and the one that we're addressing today is on the left, mapping. But de novo assembly is a really big deal as well. We're going to spend some time on that one tomorrow because it's just a hugely important technology. So, in the case of mapping, these reads that we produced, remember there are millions of them because we had millions of template sequences, our reads match to an annotation. An annotation is a term of art, and I find that new students in this field often don't really get a good grasp on it. We're going to talk a lot about annotation tomorrow, so I'm not going to steal my thunder from that. But I will just say that a genome differs from a very long sequence by its annotation. The very long sequence is not all that instructive, but once we've run things like gene finders on it and have looked for motifs and domains that we can hit in it, suddenly we have some information about which parts of this sequence would be expressed, for example. And that's one way that we can evaluate which parts of the sequence matter. So if you have a known genome, the annotation data should be pretty rich. So the human genome is pretty well known at this point. I mean, we published it, in 2001, way back when? So in the years since then, we found a whole lot of holes in what we thought we knew. We fixed those, and now I think we're in revision 38, 39, I don't even know. So we're all through time, we've gotten better and better at putting benchmarks on the human genome to say, these are genes, these are not, these are pseudogenes, etc. So given a bunch of reads, we can now determine where they match based on known sequence, based on the annotation. And that's great when you have whole genome shotgun or exome sequencing. Exome being targeted at just the protein coding genes. Assembly is really different. In this, we're trying to build contigs based on these overlaps that we see in the reads. So in this case, we know what the genome looks like. In this case, we don't. Our lab, our department, our division, has both mapping and assembly applications taking place. So this is just because you think, well, we work in tuberculosis or we work in human sequences, therefore we don't need to know anything about assembly, you are fooling yourself because there's a lot of interesting stuff going on in assembly. Okay. So why do we emphasize mapping? Why is it so significant? First off, doing the de novo route is a pain. It requires some expertise. It requires the ability to figure out, now did that work at all, or am I completely fooling myself? It's an error-prone process and it's time-consuming. On the other hand, you may be able to map a massively parallel sequencing experiment to the annotation of humans in 15 minutes. That's a very fast process by comparison to this de novo stuff. So recognizing sequence variants is a whole lot easier when you know what the canonical sequence for humans should look like, even though there are a lot of problems with the canonical human sequence. Short-read massively parallel sequencers produce more than 100 GBP per day. Is that Great British Pound? It's not. GBP, what's that?
Gigabase pairs?
Gigabase pairs, very nice. Got it in one. All right, so more than a million, sorry, more than a billion base pairs, more than 100 billion base pairs per day. It's an astonishing amount, isn't it? I said millions of sequencing reads, didn't I? If each of those is 100 base pairs, that's an awful lot of reads. Okay, so we can produce huge reams of sequencing data every time. Generally speaking, downstream, the tools that we use for kind of our advanced bioinformatics are going to assume that you've done quality control. They're going to assume that you've lopped off all the stuff that isn't trustworthy, and will assume that you've done an alignment to an annotation downstream. So to get to the interesting questions, we have to do some amount of this drudgery. Mapping is one of the most straightforward ways to turn reads into data that are aligned on our known genome. I always sort of bog down with Burrows Wheeler alignment. I would just say that the two people behind this algorithm are a little astonished that their name has become so associated with genome informatics. So if I remember correctly, Burrows was the man who invented something called the subroutine, right? He's in the pantheon of computer science gods. Burrows is right up there. The person who invented the subroutine, that is a pretty foundational thing to have invented. So in the computer science field, Burrows is not thought of as a bioinformaticist at all, and I think he's a little astonished that he's had this fame from this. Wheeler created a search engine called AltaVista. Has anyone heard of AltaVista? Oh, that's fine. In the day, I used to use it all the time. Back before Google had just sort of stomped everything else in the field, there was AltaVista, and it was really good. So these two fellows have quite an established computer science history. And part of their work was built around this concept called the suffix array. The suffix array is a rather magical data structure from computer science that allows us to figure out overlaps among a great many sequencing reads. So when you start with millions of sequencing reads, you can imagine that the software is considering the last nucleotide of each read, the last 2 nucleotides of each read, the last 3, the last 4, the last 5, and then sorting all of these together in order to find the best way to determine any intersection between sequencing reads that we produced and an annotation that we want to overlay them upon. Wild, huh? Okay, so this is an astonishing process, and I'm going to start with what my computer science algorithms teacher would have called brute force and ignorance, BFI algorithm. There are lots of brute force and ignorance algorithms out there, so I'm going to walk you through one. Imagine that for every read produced in our experiment, we ask, is this exact sequence found anywhere in the genome annotation? So how would you do this on your computer? You'd probably open the genome annotation, type Control F to open the find box, type in your sequence read, and then see if you get a hit. Okay, and your computer's like, you did what? And then it says, okay, there's a hit in gene 50, and there's another hit in gene 10,502, right? So you could do this. Okay, that's what we would call a brute force and ignorance approach to finding where this sequence, where this read aligns on the annotation. But wouldn't it be a whole lot cooler if you could build one data structure, the suffix array, that incorporates all of the reads, and then pass that data structure through the annotation once, and get out where each of those sequences hits on any of the genome annotation? That's magic. That is highly efficient, and it represents one of these places where computer science has made these technologies feasible for everyday use. So that's what you should know about Bowtie. It is the ability to build one data structure from all of the reads and then pass the whole annotation through it and find hits for any read to any gene. Freaking amazing. Okay, now what comes from that? A BAM file is very much like something that we call a SAM file, which is a sequence alignment map. But here, instead of writing it in a big text file, we're going to write the binary version of it instead. So we're going to call it a BAM file. Imagine that you have an annotation that contains 45 nucleotides. All right, so here's our 45 nucleotides. We now have an alignment, here using some software called Cigar, that has mapped a sequencing read here, a sequencing read here, one here, another here, one through here, and down here. Some of these have greater strengths, some of them have weaker strengths. In fact, in this case, we have a big gap that we need to introduce in this read to make it match properly to here and to here. We even have this really odd property that this one and this one are known to be opposite ends of the same template. This is something called paired end sequencing. Going to be a really big deal tomorrow. We're not going to touch on it today. Okay, so this is our alignment. We need to have some way to write that into a file. And under the old sequence alignment map approach, you would have each read appearing at each aligned position. You're going to have some starting position. So here we see that read 1 starts its alignment at position 7. And, let me see, right. We also, you remember we had Phred scores that represented each base call. The BAM or SAM file is also going to give us a map quality to say how reliable is this mapping between the sequencing read that you produced and what you see in the annotation. So a BAM file or SAM file, you can use them interchangeably really, is an explanation of how the sequencing experiment relates to the annotation that you have. Okay. Now, this brings us to the fact that occasionally we do not have a perfect match between the read and what we have in our annotation. I'm going to check my time here again. Okay, so I have 40 minutes. I'm going to make it. I'm not going to panic. So, I think everyone remembers seeing this at some point, right? Right, this is the genetic code. So we have codons of 3 nucleotides that map to an amino acid. Great. So now I get excited. As a proteins person, I see amino acids and I'm like, finally, we're getting somewhere. It's okay, I kind of live at one end of the food chain in bioinformatics. So we're going to talk a lot about nsSNVs. I just have to say that on occasion, you'll find that at master's defenses or honors defenses or whatever, we ask people to define terms that they use a lot. It's always worth your knowing what these things stand for. So I've broken it down into 2 parts. NS is non-synonymous. SNV is a single nucleotide variant. So non-synonymous has a couple ringers in it. There are a couple pieces to it that I want us to keep in mind. To say that something is synonymous or non-synonymous means that we're making a reference to the genetic code. And if we're making reference to the genetic code, that means that this is a coding, this is an open reading frame for a protein. Right? If something codes for a tRNA, you would not bother looking at the genetic code because amino acids don't come into it. The only time you would care whether something was synonymous or non-synonymous is if you're coding for amino acids. So in this case, I'm specifying that the DNA change must alter a codon, but not all alterations to codons, only those that specify a different amino acid. Does everyone remember the term wobble hypothesis? Oh good, they still teach under that term. I was so afraid to change the name again. Okay, so here we have CCU. Everyone remembers that U corresponds to what in DNA? T. Yeah, U replaces T in the context of RNA. So here, if we modify this U to a C, is there any change in what amino acid it codes for? Not at all. If you change U to a C at the 3rd position in UCU, you change from a serine to a serine. There's no difference from the perspective of the polypeptide that would be coded for by changing that. So if we're going to have a non-synonymous mutation or variant, we're going to have to change something like this middle U for this UUU codon to something like UCU. You see that changes us from phenylalanine to serine. Now, what happens if I change the third letter of UUU to an A instead? It's in the same box, right? So we change from UUU to UUA. Is that synonymous or non-synonymous? It is non-synonymous. Even if it appears in the same box, we see that UUU codes for phenylalanine, UUA codes for leucine. So that is a non-synonymous change. There are times when changing the third letter of a codon keeps the same amino acid. It's quite frequent. But sometimes we find that changing that third letter does alter the amino acid it corresponds to. Okay, so when we are talking about non-synonymous, we're talking about what amino acid would result from it. Typically, these single nucleotide variants are transitions. More infrequently, they are transversions. And sometimes you'll even find that people include indels among SNVs, although that's a little less common. Does everyone know what an indel is? An insertion or deletion, exactly. If you add another nucleotide to the sequence in place, it will alter the reading frame of everything downstream of it. If you have a deletion of something other than 3 nucleotides or 6 nucleotides, you can also change the reading frame of everything downstream. So indels are kind of a special case. Generally, when we're talking SNVs, we're talking about something that is a purine getting substituted for a purine, a pyrimidine getting exchanged for something that's a pyrimidine, or a transversion, where like an A becomes a T, or an A becomes a C, or a G becomes a T, or a G becomes a C. We should remember that C and T have more in common than does A and C, for example. Because A and G are these big bicyclic purines, and these are relatively small with just one ring in the nitrogen base. Those are pyrimidines. Okay, so these changes are things that we often attribute some functionality to. To say that something is a non-synonymous SNV is to suggest that this genetic variation leads to phenotype change. So what about SNVs that take place outside reading frames? Would we say they're synonymous or non-synonymous? If you're outside the coding sequence for a protein, it doesn't make sense to say synonymous or non-synonymous anymore. You may instead alter the regulation of that gene. Okay, does an amino acid change imply phenotype change? That's a much harder question. If you alter the amino acid to another amino acid, does it guarantee a change in phenotype? It does not. All right, so how do we determine that we have detected a variant? There are many different rules that we might use for this. One is that we want to have lots and lots of reads, all covering this from different perspectives, that make us very, very sure that this person has an alternative sequence at this position. If we're relying on these base calls, if they're all given with a score of 20, we're going to disregard that. But if they have scores of 40 and 50, well, we're going to feel a whole lot better about that. So having a high reliability of individual base calls really does matter. I've already said sequence coverage should be high, but to repeat it, many reads, just a high count of reads at this position is valuable. And ideally, you have some sort of test statistic to say how frequently you would see a sequence variant of this sort by random chance. So VARScan2 is just one of many algorithms that exist for doing this variant calling. Did I see a question? Okay. How can you tell then if a non-synonymous SNV is deleterious? Now, I've used the term deleterious here, but there are lots of different ways that we can describe this. Does it alter phenotype? Is it permitted under natural selection? There are lots of ways you can think about this. So I'm using the term deleterious, and let's imagine that you as a graduate student want to get some sort of score out of it to say, oh yeah, this is really bad, or no, I can ignore this, this is no significance whatsoever. So frequently we think of this as kind of a temperature gauge. This is a very tolerable mutation, or this is an intolerable one. So SIFT was published back in 2001. Its job is then to retrieve a whole bunch of sequences like this one and ask how frequently this letter is allowed at that position. So if 50% of the human population has this other letter at that position and the other 50% has a second letter at that position, obviously you can live quite happily with that sequence variant. Polyphen is an approach that was published in 2002. It adds this concept of 3D structure. Imagine that you have a section of a protein that passes through a membrane. Maybe it's got a whole bunch of alpha helices, right? You can imagine that if you add something like a proline to the middle of that sequence, it'll add a big kink to it. Something like that is much less likely to be tolerated than if you add a proline to a big loop region of that protein, because you have a lot more fluidity with what the structure looks like in that part. SNAP, published in 2007, even adds this concept of solvent accessibility. So frequently we think of proteins in solution as having a hydrophobic core, very down deep away from the outside, and then more polar side chains on the outside. Okay, so SNAP is able to use solvent accessibility as a way to evaluate that. And as is frequently the case in bioinformatics applications everywhere, we've seen that machine learning has become really hugely important for being able to separate classes of changes. So being able to discern that this variant is a very big deal can be better discriminated because we have tools like machine learning at our disposal. Okay, it's 10:30. I have a half hour of material left. But it is the intermission, so I'm going to briefly pause the camera and rest my voice. If you need a bathroom break, this is the moment. Come back and we will talk about Genome-Wide Association Genome-Wide Association Studies. So take 5 minutes, come right back. Be quick, because in 5 minutes we're going to plow. Great, so let's continue our discussion. We're now talking about Genome-Wide Association Studies. Back when I was in grad school, this was like the big grand idea that everybody wanted to execute. So I'm highlighting a paper that was published in 2005 in Science. It reflects one of the first studies where people got pretty much all of the pieces right. So the Genome-Wide Association Study helps us to address situations like this. If we can amass 100 patients with a disease and another cohort of 100 without, could we find any genetic markers, particular sequence variations, that were associated with disease risk, sometimes expressed like an odds ratio? Okay, so Klein et al., we're looking for markers of age-related macular degeneration. Everyone know where a maculus is? Right, so if you, some people as they age have the degeneration of the maculus, I guess. This causes them to lose their sight. So they thought that they might be able to find genetic changes that were specific to folks who suffered this, that were predictors that these folks would develop disease. So they had 96 cases and 50 controls. They used microarrays that measured 116,000 SNPs. Now we've talked about SNVs quite a lot. How does a SNP differ from this? SNP. What does it stand for? Single nucleotide polymorphism, good. A SNP differs from an SNV largely by the fact that a SNP has a known frequency in the population. Typically we would say that if more than 5% of the human population has this variant, it could be called a SNP. There are plenty of places where you and I differ by single nucleotide polymorphisms, and they don't reflect the fact that I'm a crazy mutant, but rather the fact that I just come from a different part of the human family, right? There are SNPs on which my brother and I differ as well. Okay, so SNPs are not really disease causing in their own right. They more or less function as benchmarks within the genome where people are known to vary from each other. So they found 2 SNPs out of this collection that associated with the disease, and suddenly everybody was publishing Genome-Wide Association Studies, or GWAS. Okay, one of the key technologies that they put together was a visualization of the data they had produced. So this is the Manhattan plot, and you can see that we have basically a very high density bar plot, right? Each of these is representing a particular SNP, not a particular gene, but a particular SNP. And we see that some of them have very high bars and some of them have very low. Now, I want you to think back to what we just said about Phred scores. You remember that Phred scores are the negative log of a probability. The probability here says this variant is distributed at random in this population. So if you have a high bar, the statistical test is saying that it's very unlikely that this gene is split randomly among these two cohorts. It's not saying this gene causes this disease. It's really easy to make that mistake, especially when you're very early in grad school. We like to think that science gives us beautiful answers like that. Instead, these p-values are evaluating a null hypothesis to say this gene is randomly, some people have A here, some people have C here, and it has nothing to do with this disease. That would give you a very high p-value. So what we're trying to focus on are those few SNPs that really do seem to associate very strongly that everyone in the case cohort has a C and everyone in the control cohort has a T. Something like that drives the p-value low, and as a result you get a high bar in this chart. The lower the probability, the higher the Phred score. The lower the p-value, the higher the bar in the Manhattan plot. Why is it called Manhattan? Is it because they were from Manhattan, Kansas? No. No, Manhattan, Kansas is close to where I grew up, actually, but the skyline of Manhattan has a whole lot of towers, so a nice Manhattan plot is going to have tall bars to reflect these particular single nucleotide polymorphisms that associate strongly with the cohort separation. Okay, so the negative log of p-value is the height, and here we see a really important early demonstration of multiple testing correction. I'm a freaking Nazi where multiple testing correction comes into play. It is the number one thing I ask people about at their defenses, so we're going to talk about it a lot more on Wednesday, but I just want to point out that this is one of the first places that people brought it into play. How many different SNPs were they evaluating? 103,000. 103,000 SNPs. Just for reference, how many genes do we have? How many protein coding genes in the human genome? One of my favorite questions to ask. About 20,000 is the number. So we have about 20,000 genes, and here we see that they are looking at about 103,000 genetic markers within the genome. So we see then that we could ask, if we were looking at a p-value of less than 0.05, how frequently should t-test give us a p-value below 0.05? You just had statistics, so I know that this is fresh in your mind. How frequently does t-test give you a difference, a p-value below 0.05 when there's no real difference to be observed? 5% of the time. 5% of the time, t-test should give you a p-value of less than 0.05 even when there's no difference. So if you're testing 100,000 markers, what is that divided by 20? 100,000 divided by 20 is 5,000. So even if there were no differences to be observed, you should see a p-value of less than 0.05 5% of the time, which means that 5,000 of these markers should be significant p-values if you're just using the rule, is it below 0.05 or not? That's why they knew they had to be more strict than that in judging whether something was a hit or not. So they decided that they would use an approach based on an Italian mathematician's work. Bonferroni was an Italian mathematician, but actually a female biostatistician realized that his work was really applicable in the context of statistics. So she put together this strategy where she said, if you know that you're going to perform 100,000 tests, divide the threshold you want by the number of tests you do, and use that as your threshold. Clever, huh? So now we see that this line right here has been drawn across the Manhattan plot. It's a very important line. What they're saying is, we can't use 0.05 as a threshold because we are performing so many bloody tests. Instead, we need to raise that threshold to a much more stringent level by dividing 0.05 by about 100,000 to get our new significance level. So these skyscrapers down here, they don't matter. The only ones that matter here are these two that happen to be above that line. So they were much more restrictive about which p-values could be considered significant. They still had these two markers that passed that goal line. Okay, so that's how to read a Manhattan plot. Don't worry about this fuzz down here. It doesn't matter. Where this bar is up here is what matters. Are there questions about that? Everyone still has a warm feeling in their hearts about biostatistics after that last week, right? That's good, that's good. All right.
The exam yesterday did not go well.
I heard the exam was tough.
Yeah.
I hear it every year, actually. The guys who teach that have put a lot of effort in this year to try to make it more approachable. It may not have fully succeeded, but it's the first time they're doing it this way. We'll see. All right, so let's discuss one of the big challenges we can make to this. We generally think that each of the tests that we're applying when we do a whole bunch of statistical tests are independent of each other. Independent and identically distributed, do you guys cover that term, ID? Well, a lot of times people assume that each of these markers that they're testing have independent value, are independent of every other finding. But in fact, what happens when you have two different genetic markers that are quite close to each other in the genome? If someone has SNP variant 1 at this position, the neighboring marker is also quite likely to be SNP variant 1 because these co-segregate, they show up together. That means that statistically testing each of these as an independent entity is already kind of a mistake. So linkage becomes a very important property in this. See all these little RS numbers? These are a type of address and a type of accession that we use for these different SNPs. We see that all of these across this 38 kilobase region, they all fall within 38 kilobases. That's close, right? All of these have these red values here to show a very high degree of correlation. So having tested marker RS1535, and you can already have a very good prediction of what the value for RS174549 is. So linkage really throws this concept of statistical independence into the hazard. So the concept of linkage really then matters. And frankly, we're relying on this in a lot of ways. Because remember, I said SNPs are not generally considered to be causative of disease. Most of the time, SNPs don't fall within the open reading frames of these proteins. Instead, they happen to fall near them. And so people are suspecting that whatever mutation is hypothesized to cause this disease is in some gene, probably, that's close to where this marker associated with the disease is. Which leads to this big problem. If someone says these two markers are associated with this disease, how do you translate that into a cause? A cause. That's much harder, isn't it? The typical thing that you see people doing is to say, this marker is located at this genomic coordinate. There is this protein coding gene that's 50 kilobases this side, and we think this person has a mutation somewhere in that gene that is causing this disease. And that's a lot of supposition. And really, until they've gone and sequenced a variant that they find in association with that polymorphism, it's very hard to make that kind of claim. So GWAS frequently struggles to get to a place where they can say, well, we know this marker is associated with the disease, but we really have no idea what causes the disease. Okay, PLINK, or PLINK, is some software that is very, very widely used for GWAS data analysis. I've included a link to it down here. In the most recent versions, they've done a lot of changes to data management. You see, scaling is a really big issue. As we've moved to the ability to do full-on sequencing to find every possible sequence variant that this person may contain, the number of, the amount of data that we have to sift through to make our assessments has grown very large. It's much easier to do stuff like this if you just have microarrays. Being able to do summary statistics through parallel computation matters. Parallel computation matters because almost every computer you buy today, almost every desktop computer you buy, has at least 4 processing cores. If you don't have parallelized code, you're not taking advantage of all the capabilities of that CPU, and frequently you're using less than 25% of its possible throughput. Population stratification is a very, very big deal. We're going to talk about its effects as we talk about admixture particularly in just a slide or two. Association analysis, of course, we need to be able to say which variants are associated with our phenotype of type. And they've done quite a bit of work on identity by descent estimation. So if you have people who are related to each other in your study, then not only have you got a problem in that the variants you're calling are correlated with each other, people from the same family share genetic material. So again, now your cases and controls may not be independent of each other. Okay, but GWAS is frickin' popular. If you go to the GWAS catalog, every one of these little colored circles, diamonds, whatever they are, represents a genetic marker that has been associated with some crazy thing. That's a lot of GWAS studies, right? Which means that if you are working in some disease and are thinking of doing a genome-wide association study, it probably behooves you to take a look to see what the findings were of somebody who worked on it before. We're going to talk about some of the mechanisms for doing that this afternoon, the gene expression omnibus. All right, so let us talk a little bit about stratification. This is the part of the lecture where I always feel like I'm stumbling all over myself, so I hope you'll pardon me if I do. What if you have a systematic ancestry difference between cases and controls? So I've got an example here. Let's just say that I'm studying asthma. I'm just picking a phenotype at random. If I'm studying asthma, and I find that 60% of the people who had asthma in my study come from Han Chinese ancestry, but my controls are only 40% from Han Chinese ancestry, it could be that the markers I identify as being associated with asthma are the same markers as are associated with Han Chinese ancestry. Do you see why? Because the fraction of people who are Han Chinese is 50% higher in my cases than in my controls. Now, does Chinese ancestry have anything to do with asthma? Probably not. Probably not. But I could fool myself with my own data if I don't take that into account. So frequently, we must use ancestry informative markers, AIMs, as a way to make some batch assessments of which genetic factors are stratifying our cohorts in this way. There are people who've done quite a lot of work with the South African colored population. Used to be their poster was right out here. We've now changed that hallway, but there's been a whole lot of work in trying to untangle the complex family histories of people in the South African colored population, because where their ancestors come from can have quite a lot to do with which diseases they're susceptible to. What if you have, what if we were to assemble cohorts in studying diabetes and ignore the fact that some of the folks have strong Indonesian background and others have less? Right? That's a factor that could lead to a stratification in our data and cause us to come up with the wrong conclusions about association. So admixture is something built on this concept that whenever you're dealing with genome-wide association studies in populations that have this complex mixture of ancestry, you have to take the fractional contributions to their ancestry into account. Now this became a really big deal in the United States because people correctly apprehended that people were not doing a lot of studies in the African American population. And if all the GWAS studies are performed in the white population, how much you can extrapolate from there to the African American community was basically unknown. But in the African American population, there's a huge variety of African populations that have led to the black part of their ancestry, and frequently you have some amount of white ancestry mixed in alongside that. So being able to untangle those roots is quite important. We have until 11, I believe. So what we see is that if we are to look at people from the South African colored population and ask where are their markers reflecting their origins, we find that we can sort them in any number of ways. So in each of these graphs contains exactly the same people. But in the first one, we have sorted the individuals based on how many of these markers reflected what are thought to be the Khoisan variants at these locations, the Khoikhoi or San. So we see that some folks in the colored population have next to no ancestry from the Khoisan population. Others have a nearly pure reflection of what we think to be Khoisan variants, Khoisan alleles at these markers. If you were to look at populations that we think of as African, and here we're having to draw from lots of different groups. I mean, Yoruba is hardly representative of what you would expect for people from Black African populations in South Africa. We see that some people have, within the colored population, have 0 Black African alleles. Others have nearly perfect representation of what we think of as these forms. And the European contribution to folks can also scale quite a lot. The South African colored population represents a meeting of people from all of these different parts of the world. If you think about the Asian contributions, certainly we have a lot of evidence. I mean, we tend to call them the Cape Malays, but in fact, a lot of Indonesian populations gave rise to the South African colored group as well. If you simply try to pursue a GWAS without taking ancestry informative markers into account, and admixture is an important feature of that population, you're going to have a really big mess on your hands, and you will make the wrong associations of understanding which markers are associated with that phenotype. So this area, that of admixture relationship, is a really important one in being able to perform GWAS in groups that have been historically less studied than they should be. So, it's tempting to say that since there's a population with its roots in Europe that's been studied for some phenotype, that we already know all the markers that could possibly matter for that phenotype. Americans, white male Americans, have been making assumptions like that for a bloody long time. And yet we know that there's an awful lot of genetic variation in African populations that we know next to nothing about. So you will very frequently see grants coming out of South Africa that talk about building this base of genetic information so that we understand what genetic variation in Africa relates to phenotype. Okay, so sequencing experiments are analyzed through a cascade of bioinformatics steps. There are lots of different pieces. You will frequently be using whole pipelines of these tools from FASTQC. Phred is something that's output by the sequencer. You're going to be using trimming, you're going to be using mapping, you're going to be using variant scanning. Many of these things all fit together in these long pipelines of tools. Error can creep in everywhere. If you have a problem early in your pipeline and are allowing that error to propagate forward, you may find that the results that come out at the end of the study mean nothing at all. That's a hard place to be. Statistical considerations are inextricable from bioinformatics processing. Almost every bioinformatician needs to know a lot about biostatistics in order to make sense of it. This is one of the reasons why we emphasize biostatistics so strongly in this honors program, because you're going to need that knowledge to contextualize the findings of your experiments and to protect you from yourself. Richard Feynman once said, "The easiest person for you to fool is yourself." And I do not want anyone in this room to be tricked by their data into believing something that's not true. Imagine chasing for 3 years of a postdoc after something that was an erroneous conclusion from a PhD study, right? That would be rough. So, statistical considerations matter. The massively parallel sequencer may have eclipsed Sanger sequencing, it may have eclipsed microarrays, but both may be used to confirm its findings. You will frequently see that one of the things used in massively parallel sequencer driven research is that people go back to confirm sequence variants by Sanger. Having orthogonal methods that operate differently to confirm each other's findings makes it much harder to be misled by a chance finding.